Hey guys, welcome back to Bioinformatics with R. Today we're going to get into some actual bioinformatics data. We're going to get into some microarray data. As I said in the previous video, microarrays are a bit outdated and they're not really used much anymore, but they still have value because that's a lot of data that's online that's easy for you to access if you want to do meta-analyses or things of that nature. Isaac, how many times have you tried to record this video? Too many. We've had some technical difficulties throughout recording this, so hopefully it runs smoothly. I had done these analyses a long time ago and the packages have been updated in compatibility issues, but that should be all taken care of on the PRAXIS system, and hopefully all taken care of on my system as well. So let's get into it. Alright, so the first thing you'll notice here is that we have a whole bunch of libraries that we have to load in to run these analyses. So the first thing you'll see here is this Arabidopsis database library. There's actually two of them. I'm going to put them together here. So both of these are Arabidopsis database to give us some gene annotation and some additional information about each gene that we find differentially expressed. Same thing with the annotate package, it kind of works with that. Affy is a package to run Affymetrix data. Affymetrix is a specific type of microarray, so depending on the actual maker of the microarray that you used, you'll have to use different. Agilent is another one, but Affy seems to be, I think, probably the most popular microarray. Lima or Lima, I've heard it pronounced both ways, that is a package to do regression analysis to find differentially expressed genes on microarrays. Oligo kind of partners with that. dplyr, you remember from data wrangling, where it is to kind of reshape your data. The other thing, reshape, is similar, but it's not part of the tidyverse, and since these are older analyses, the tidyverse wasn't as prevalent as it is now, so we'll have to use some reshape. Hey guys, I'm making a quick edit to this video. If you look at your packages over here, you'll notice that Affy's not installed, even though in the video it calls for calling the library Affy. So what you're going to have to do is install it real quick. IOC manager install Affy is the command. So you run that and it's going to start the install. It might take a few seconds. And what's going to happen is it's going to ask you if you want to reinstall all the dependencies, and for some reason it seems to kind of hang up when you do that, so you're going to click N for none, and then you should be able up here to call library Affy, and it works. So let's see if I update this. Oops, no, I don't want to do that. If I click to files and then back to packages, actually let's do this. Quit session, start new session. You don't have to do this, I'm just double checking that it's installed. So did call it when I did it, but it should also show up in the packages, so I just want to double check. Affy is installed. Beautiful. Okay, so that's how you get the Affy package installed. Everything else should be installed. I didn't have a problem with anything else. If you're having an error, try reloading all your packages. So run, there's the text, we have, let's see, Affy, all these other libraries that were up here, was Affy.io one, etc. So run that command again to reload all your packages and everything should work fine. So I'm not going to re-record the whole video. Continuing on with the video, that should solve your problems. So the first thing we're going to do is we're going to read the cell files. So you should have the cell files in your My Files or your working directory, the same directory that you save this. Let me go back real quick. So I'm setting my working directory here to where my R script and my cell files are. For you, if you're doing this on PRAXIS, I have the code here, set working directory, desktop classroom, My Files, and that should be where your cell files are located as well. So we're going to read cell files into the directory, but first we're going to do our targets. So our targets, we have 6 cell files, 3 flight for plants that were grown while they were on a space shuttle, back during the space shuttle era, and 3 that are ground control. So we're comparing how gene expression changes when you're in space versus when you're on Earth. So we need to tell R which samples are which. So you should have a file that you copied over called brick16 targets.csv. And it's a comma separated file, CSV, so sep equals commas, so it knows how to read it, and then row names equals file name. So we're going to convert the file name column to the row names. So let's run this real quick. And you see that we have the row names are the same as the file name here, and then we have our gravity condition, whether it's ground or flight. And now what we're going to do is we're going to read AFI. Let's call this variable AB. So we have an AFI batch here where you can see this is just all the AFI data read in from those cell files. So we'll start to process it here in a second. Okay, so let's look at a histogram quick of these microarray experiments. So I'm going to zoom because it's under my picture here. So as you can see here, we have the 6 experiments, and this is the intensity of expression. So remember, microarrays, they use a hybridization method that has fluorescence, and so they look at how brightly something shines, and that quantifies how much of the RNA is present, okay? And so this is the intensity of the brightness or the expression. So the intensity is really the count of the protein or the RNA, right? And then we have the density. So this is showing per experiment kind of the average intensity over the entire experiment. So you can imagine that there could be some sort of error based on the chip or based on the RNA prep, where some experiments just have a lot more RNA than others, right? And so this can be an issue. So what we have to do is we have to normalize the microarray experiments. All right, so we're going to normalize the microarray experiments. So we'll do this, a new variable called eset, which stands for expression set, and we'll use the AFI package and RMA, which is let's see if it comes up. That says what it is. Let's try this again. It popped up for a second. Here we go. So RMA is a function that converts an AFI back object to an expression object using the robust multi-array average expression measure. So it's a way to normalize these, and we'll see how that looks in a second here. So we're going to run this. So it's background correcting, normalizing, and calculating the expression. And now let's plot the data, P data for ESET. Okay, actually, so to run that function, now we're going to plot the data. Okay, so we're going to do histogram of ESET. Let's actually annotate our code. So let's visualize the normalized data. Okay, now let's run it. Okay, so now if I zoom in on this new graph that's under my picture here, you can see that compared to the last graph, it's a lot tighter of a fit of these experiments over one another. So let me go to the last graph here. So you can see there's a lot of, like, here's the peak for one experiment down here, and here's the peak for another experiment. Once we've normalized it, we get, you know, all the peaks are in this little tiny area here, and it's a nice tight fit. So we can compare across microarray experiments after we've normalized like this. Okay, so let's go down a little bit here. So let's kind of let's characterize the data a bit. Okay, so first thing we're going to do is we're going to create a variable called IDs, which is the feature names of ESET. So if we click on, so if we load, we'll do head because we don't want to load everything. Head of ID, you can see that these are the probe names for each gene in the on the microarray. So symbol is another variable that will say get symbol, which is from the annotate package, of ID. So what we're saying is take all these IDs, look in the Arabidopsis database 1121501.db and get the gene symbol for it. So if we run that, now if we say head symbol, you can see now we have the probe name, but also the actual gene symbol. So if you read like a scientific paper or whatever, this is what generally they'd use, right? And now let's do affy data, a new variable, and say read dlim, and we're going to use this ath1 array elements file. So this should be one of the files that you copied over. This is just a kind of a data table of a whole bunch of gene annotation stuff that comes from TAIR, which is the Arabidopsis Information Network. It's kind of the consortium that publishes the genome and has a bunch of information on just Arabidopsis, and so they publish this file. So we're going to run this, and so if we look at affy data, you can see we have the element name, so the probe name, what type of probe it is, so oligonucleotide, so it binds to RNA or DNA, the organism, Arabidopsis, the locus, so this is the actual gene annotation for like the genome, the description, so what kind of gene it is, so an NADH dehydrogenase subunit 4L, the chromosome, so this is M is mitochondrial, C is chloroplast, and then when we get to numbers, these are actually like the chromosome number, right? We also have the start and stop location on those chromosomes, which is kind of cool, and that's about it. So gives us a lot more data to work with to actually annotate and make biological sense, because that's one of the big issues with bioinformatics, is that a computer scientist could do bioinformatics, but the issue then becomes how do you interpret the data and put it into a biological context. And I actually had this conversation with another faculty member just yesterday, talking about, computer scientists, they have all the tools, and this is not a knock on computer scientists, it's just a different mindset. And they have all the tools and they can do all the coding, but you have to be able to take what is spit out of your analyses and then make apply it to biology, right? And to say this makes sense or this doesn't make sense. And so this is going to give us some more information on how to turn our analyses and answer biological questions. So the next section here is we're actually going to do differential gene expression analysis. But for sake of brevity, and I know y'all like short videos, I'm going to keep it short here, and we'll break this down into another video. So we're off to a good start. We've got all of our data loaded in essentially. What we have left is we're going to run the differential gene expression analysis and then make some biological sense of it in the end. So I hope you enjoyed this video, and I will see you in the next one.